in here. So.
Okay.
Just okay, thank you.
Is it full view? Where is full view here?
Well, this vista, yes, modalità
Schermo intero, schermo
This
intero.
one, okay.
Control L.
Okay, great. So we start our plenary talk. We are a little bit late, but the coffee break was so abundant that people have decided to stay there longer than expected. It's my great pleasure to introduce Sylvia Richardson. It's also a great honor, and I thank the organizers for asking me to chair this session. Sylvia Richardson has been recently elected president of the Royal Statistical Society, and those things don't happen by chance. It has happened because Sylvia Richardson has done very important research all over her career. She is now a professor of statistics and director of the Biostatistic Unit at the University of Cambridge. Before that, she has been chair of biostatistics at the Imperial College in London, and before that, she was director of research at the French National Institute for Medical Research. Her research has focused on many different areas, all related to biostatistics, and she has given important contributions to the field of statistical modeling, particularly from a Bayesian perspective. She has dealt with spatial modeling and disease mapping, measurement error problems, mixtures and clustering methods, and she has recently devoted her attention to the study of large data problems. I am personally indebted to Sylvia Richardson because after reading her paper on matrix sketching, I've started my current line of research, and so she has given me the idea for nice things that I'm doing now. So I'm particularly pleased to welcome her. The title of her talk will be Statistical Challenges in the Analysis of Complex Phenotypes in Biomedicine. Thank you very much.
Thank you, Angela, for a very nice introduction. Thank you to Francesca and the scientific committee for inviting me, and to the local organizer, particularly Giovanni, for making this stay so nice and so, all the arrangements perfect. So, Angela has introduced, the topic of my talk, which is focused on exploiting in the area of statistical genomics, and in particular trying to make the best use of these type of data, which is now being collected throughout the world on patients, on people, also on healthy patients, to try to understand better, basically, the origin of disease and really the biology of the process. Now, so this has been the motivation behind our work, and part of our unit. It's one of the 4 main themes in our biostatistics unit. Now, of course, many of you don't work at all in that area, and I just hope that some of the modeling ideas that we were confronted with will, there's the generic issues, everybody can translate or just find some connection with their research. And so basically I will talk of 3 different aspects of this research to broaden, not being too technical in each of them, to sort of show the type of ideas that we have been developing. So as I was saying, there's a big expansion of these expensive data being collected, which tries to characterize really in depth the molecular characteristics of our own body. It's either focused on some diseases, for example, cancer or rare disease. So in England, there is the 100,000 Genome Project, which is really doing full DNA sequences of 100,000 patients to try to characterize better their disease and then possibly their treatment. It's also, so there this data would be used to look for rare variants, for example, in the case of disease, and I will have one aspect of my talk is focused on rare diseases. Also, you can just try to follow up the general population. So there's this massive cohort, which is now, the idea is being replicated in Germany, in the US, where they want to do 1 million rather than 500,000. And again, these people have deep phenotyping, but also a lot of measure on their health and well-being and so on, environment. So there's this massive increase of health information, and of course a lot of our statistical background and a lot of the statistics we know can be used to tackle these, but often we need to develop, to tailor this approach or develop novel approaches. One thing that probably all of you have heard about is these genome-wide association studies, which are incredibly a simple type of design. Basically, you measure variation along your DNA code, and typically when I talk of variance, it could be it is as simple as substitution of one base pair. There's variation among the population. Most people would have one base pair, some people would have another one. So at the level of just this one letter, basically, then this is linked. It is just a massive jump, and it's linked to maybe people having that particular substitution would have a tendency to be to have a disease, cardiovascular disease or some other disease. And so these GWAS and the search for these meaningful substitution variants have been going on for a number of years, and there's a catalog of all these variants which have been reproduced and which are, people have confidence. The GWAS catalog, I've said 6,000 of those, but every day if you look, it goes up and up. The problem is that even though we find this position, most of the time they're not within genes, so they're not within what is known in our body to do the protein that we need to function, and so it's very difficult to interpret them. And they also have a statistical significant effect, but weak effect, so they're not the major, on their own, the major driver. So to try to understand a bit better what's going on, people are now measuring the whole path, are trying to, because there's the biotechnology as developed, are just measuring the whole path, basically, of the biological flow of information, basically, between the DNA code and what we capture, at the very macro level on our health. And in that path, the first step is what's called the RNA, so it's a message, it's a copy, a single-strand copy, of bits of the genome, and that's going to be used by the cell to produce this protein. So there's transcription, translation, the protein, they form complexes, they go inside, they go together in some pathway. At the end, it's what we can capture at a macro level in our blood and so on. So this is really complex, and along that, there's lots of different types of data which can be collected. And now it's the attention has focused not just to go directly from the DNA to the disease because it's limited understanding, but to try to capture that pathway, and so to model multiple layers of information. So now, in terms of what is the statistical task for that, as you, these GWAS were really single one variant and one. This is they were extremely simple statistics, p-value, odds ratio, one at a time, and massive multiplicity correction. So there was quite a lot of work on how to do this multiplicity correction, but apart from that, the statistics is very simple. Now, as we know here, you could exploit the data better by using multivariate analysis and using not just one at a time feature, but groups of features together. Of course, when you do that, you need to account of the uncertainty in how you extract features. So that's one of the task we have to do. And as I was saying, rather than going straight from the DNA, try to make use of these multiple responses, these multiple layers of characterization to get what I call richer, more complex phenotypes. So do joint analysis, for example, joint regression analysis will be one part of my talk, of multiple quantitative traits, or developing model for clinical data for rare disease. Often it's like a series of symptoms which are refined, has this patient got this? Yes. If yes, is it an anomaly of this or that? Some kind of discrete classification of symptoms, and how do you treat that as a response data? And finally, sometimes just to get the first broad view of what's linked to what, you need to just test for conditional independence and do a kind of efficient kind of network reconstruction. So these are the three aspects I will talk about. And here I want to say that our focus is not on prediction, which is also very important, but it's focused more on understanding, so finding feature, finding links, and so on. Now, in terms of, as Angela was mentioning, for a number of years now, I've worked, and I find that the Bayesian framework is particularly powerful for this kind of complex biological, medical phenomenon. And so one of the ideas to try to build joint models, so exploiting, together the features, but accounting for the uncertainty, not try to home in on the one model, but for example, benefiting from model averaging. Now, when you have very many features, molecular features, and you have large samples now, but they're still not huge, and in particular, even though the GWAS samples are very large, the samples where you've got this detailed characterization of the flow information, because these are very expensive, they're much smaller, so they're a few hundred, they're not, in the 500,000. So you need to use sparsity when you want to extract some signal. We are in the realm of using, saying that we expect only we want to extract a few important features, and how do you actually specify this sparsity is challenging, and seeing the influence of that. You need to borrow information, and we'll see that particularly in the context of rare disease, where there's a few people, nobody looks like another patient, and so how do you find some kind of similarity between the patients to actually still pull information across them, and I'll talk about that. And then if you want to use Bayesian method, there's the problem of scalability, and simulation-based algorithm, MCMC algorithm, become notoriously slow, and then so either you try to be in a simplified, whether you could do actually, you could compute everything, you have closed form expressions, which are then become very fast to use, or you try to have approximation rather than having the full posterior, for example, using variational inference. So these are the three I've already basically described. I'm going to talk about three linked topics: this hierarchical related sparse regression, where we're going to have a common set of features but many responses, and it arises in the concept of the transcription. I'm going to talk about something which we've developed called phenotype similarity regression, where we try to find common similarity between groups of very rare patients, and to try again to associate that with some feature variant, rare variant. And then the framework of trying to find basically the conditional independence, the direct association among a very large number of features. So in terms of the first topic, two colleagues are absolutely crucial in this work. There's Hélène Ruffieux, who did her thesis in Lausanne, and with Anthony Davison, and who is now working in a unit for a couple of years, and Leonardo Bottolo, who is a reader in the clinical school in Cambridge. So I was saying the statistical framework here, and I'll talk about the biological motivation in a bit more detail, is basically you have very many quantitative traits, and you have a large number of predictors, but these predictors, because there will be the DNA, they're common to all, so it's the same set of predictors, and then you have many, many responses. So Q is the response, P is the size of the predictor. So we've got basically, if you're trying to select the association, if you try to do this regression and select significant association between predictor and the responses, you've got this large Q by P matrix of selection indicators. And you will need to actually encode some structure about, first, you're not expecting many association in that respect, but you also might be expected that some predictors are influencing several responses. So you might be expecting some links across these responses. That's the whole point of taking them as a joint block. And you still want to be scalable. So as I was saying, this arose from this first, I don't know if you recall that little cartoon I had, the first process, this process of transcription. So to try is the first instruction given by the body, basically, from the DNA, which is copied. Then many, many quantities are transcribed. You could look at all the genes, roughly 25,000 genes, 20 to 25,000 genes, and at any instant, this is a movable, whether the DNA is fixed, these instructions continually change. But you can nevertheless capture them. So you're basically capturing 25,000 transcription measure when you actually, use the right technology. And so some of these, so you are in a region of the DNA. In there you got all these variants, so these possible substitution of base pairs. Now, some of these are known, genes are very well known where they are in the genome. So some of these variants will be within one of several of your, physically within these genes, which you are looking at the transcription. And these associations, so if you actually perturb the chemical there, then maybe that gene just cannot function. So these associations are quite strong, and they're easy to find. And typically there's the one which you find by just simple, p-values, test of association. And these are called cis effects. But there's also, it's now known, but it's harder to detect, that a variant here within a gene can influence the expression of many of genes which are very far away physically, in the genome. And these are weaker effects, and they're called trans effects, because they are not within but outside. And there's great interest in trying to detect some sort of key hotspot variants which would be associated with many of these responses. So this is the motivation for analyzing these 25,000 responses all together with the predictor. Then these hotspots are hard to detect because basically they get, if you just do one at a time thing, you just get killed by multiplicity correction because they're weaker. So we have developed a sort of hierarchical model to actually share some information across all these responses. And so this hierarchical related sparse regression, which is a generic way of linking, many responses to the same set of predictors, if you think there is something, some connection, something to borrow, it is tailored to the detection for these hotspots, so these predictors which would link to many of these variants. So it's multivariate, if you want, in two ways. First, it's because you've taken a whole region, say 2,000 or 5,000 variants, a block of variants. We cannot run genome-wide for 300,000, but we can run for a few thousand predictors. And it's looking among that region, because there's very many, these variants are often very correlated because of evolution. And so it's looking which are the likely set multivariate in that way, but it's also, you'll see in the parameterization, it's also, we're also introducing a common, if you want, a random effect, which is common which is specific to the variants, which will be shared across the responses to be able to try to detect these hotspots. And to make that scalable, we use variational inference. So this is the picture of the model. So it is, T is going to be the response index, and S is going to be the predictor index. So you've got P predictors, Q responses. So you first have the very common formulation of spike and slab to try to actually, regress one response on the set of predictors, but assume you've only got a few of these predictors. So the coefficient beta ST, so there's a whole matrix of the coefficient beta ST is either 0 with probability 1 minus gamma, or it's non-zero, and in this case it's given a normal prior with a variance, which is decomposed in terms of a sort of a global scale. And then, because some gene expression are very variable, some are not variable, so you have this tau T, which is a specific response variance. But what we're focusing on is the sort of the modeling. So you've got this underlying selection probability, this matrix, and there's the modeling of those to be able to borrow information. So how do we model those? Well, as I was saying, you want to both control sparsity in each of these regression, but you want to borrow something. So it's quite natural and computationally advantageous to go on the probit scale for these 0-1 indicator and have one random effect which is shared. So it's like a propensity of a predictor to influence many responses. And then you've got a parameter which is associated, the parameter tau T, which is associated with each of the responses. So far, so good. Now, the difficulty there, the sensitivity there, is how do you fix the variance of these effects? And in these types of setting, you have to watch. Variances are the crucial things, and anybody has to sort of be aware of that and really justify how you choose these variants and how you try to let the data speak, but put some information. And here, what we have used, so I think the novel aspect is that for this propensity, this variance predictor-specific random effect, we have found that the variance of that can be really influential. And when you have many, many responses, if you fix a variant, sometimes you just have a pile-up of stuff. It just suddenly, finds a little bit of information, and there's nothing to take it back to reasonable values. And so somehow that variance needs to be linked to, in a sort of flexible way, to the number of responses. And we've used something which is called the Horseshoe prior, which I'll detail in a minute. So first, if we look at the other component, the variance, the specific bit of the Xi-T, which is linked just to the response, then it needs to adapt to the sparsity level corresponding to each response. So it really is going to depend on what's your P, how many predictor you've got, and what are you supposing in that region. Roughly, are you expecting 1 or 2 basically hits, or are you expecting many hits? It will depend on your situation, and so you could choose these hyperparameter to match that. But for the variance-specific random effect, the theta S there, we have used what's called a horseshoe prior, which has now been well studied in the case of just sparse regression in general, but here we're not using it on the beta, we're using it on the selection indicators. Because what you want is basically, this variance will have a shrinking effect, but what you want is, in a way, shrink most of the, most of these variants will not be hotspots, and you basically want this probability, to be very small. Some of them will have large effect, and you don't want to shrink them unnecessarily. And basically, this specification with this product of this lambda, which is a feature-specific bit of the variance times a global penalty, the sigma 0, has been found to be very effective for allowing both controlling the shrinkage through the sigma 0, which is common to all the predictors, and the lambda s, which is given a half Cauchy distribution, which basically has a large tail and sometimes allows no shrinkage. Well, I've already explained that we've got these product things, and to illustrate why is it called a horseshoe, it's not because the Cauchy looks like a horseshoe, it's because if you look at the actual shrinkage effect, how much the omega will be they will be put towards either 0 or towards 1. So it's the actual overall shrinkage on the coefficient, which has this kind of U shape, which can be either 0, which is most of them disappear, which is the sparsity you want, but some of them don't get shrink at all. And analytically, if you look, that's what happened. But what we found by looking carefully at the effect here of the effect is on the number of responses, is that we need the scale of this Cauchy, half Cauchy for sigma 0 to be inversely linked to the number of responses. So we have a kind of a justification for a sort of automatic adaptation to the number of responses that you're using. In the order of speed, I won't. So coupled with that, so this is the model, and I try to explain, the key feature of this modeling of this matrix. Now, coupled with that, we've developed, Hélène's developed variational Bayes version, i.e. an approximation of the posterior distribution. So it's not the full posterior distribution. It's an approximation where you're assuming a factorization in terms of the parameters. Of course, the betas and the gammas, the selection and the betas are highly correlated, and it'd be really stupid to have that as the product. So obviously this is kept as one joint distribution in the product, and it's not. Otherwise, the rest of the parameters are a product. And nevertheless, as people know who have actually, anybody who's done regression with highly correlated predictors, know that you get stuck sometimes. You find one, you converge with one explanation, and alternative explanations are not explored when things are highly correlated. And so we find that it was the algorithm was much improved if we had, instead of going directly for the optimization, just doing some simulating annealing, sort of reducing the effect of some of the modes with a sequence, and then going through that. And to illustrate the algorithm, this is a simulation where the P is 200 and Q is 20,000. And there's strong collinearity in some of the regions, and only 400 samples. And here you see, so some of these hotspots, there's 5 hotspots associated some with so the actual value, the true value is where the little crosses is where the number of responses, so some were associated with 100 responses out of the 20,000, some were less, and you see very good recovery. But if we're not using simulated annealing, you could see that you still see the hotspot, but the size, you get stuck along the way. So it is really important to actually think about, like you think about the mixing of the MCMC here, you need to think about the optimization, the fact there's multi-modes, and that you need some flexibility to explore it before you converge. This is a real example, which I'll come back at the end, where we've used some expression data, not on human, but on culture cells connected with the immune system, and to try to understand, how the immune regulation, and they're in 3 regions which are known, so 3 regions of about 1,500 SNP under different conditions. We find sometimes no signal, and for one of the region, for one gene, this Liz gene, which is a gene which is has an influence against a bacterial infection, in getting the immune system working against basically bacteria. There's evidence, as you could see from this the one on the right there, as the division of this column, that under several of the conditions, unstimulated and stimulated, there is evidence that it's linked to quite a few number of genes, like 300 out of the 24,000. And I'll come back. And this makes sense. We collaborating with the team in Oxford, we're producing the data to interpret these hotspots. And I want to move on to the rare disease framework. And acknowledge 2 colleagues, Ernest Turo and Daniel Grimm. Sorry, their names didn't appear. They appear at the end. Daniel was a PhD student of Ernest and I, and he's now a fellow. He's got now a fellowship in Cambridge, and Ernest works in the hematology department. And so here are we're working with a group in the hematology department who are really interested in rare blood disorders. And so BPD is blood and platelet. Platelet is what makes your blood coagulate. Platelet, blood and platelet disorders, rare diseases. Now, when you have patients with rare diseases, typically they have many symptoms, and basically the way to characterize this patient is collect the symptoms. And for some, and then throughout the world, to try to improve, exchange of information, people have agreed a standard way of characterizing these symptoms, so standard ontology. And the basis is if you have standardized how you characterize this patient, you have more chance maybe to find the rare variant which could create these disorders. And even though these are rare diseases, it's important. Rare diseases are like human model of what you can't do. You can't do an experiment where you actually knock out something and it kills the person. But the rare disease, in a way, do that themselves. They are serious perturbations, which have, most of them have been eliminated through, evolution, but some are remaining, or some are de novo, and exploiting that can really give us this kind of unfortunate, close to an experimental model. So it's important to make use of that information. But of course, no 2 patients are quite the same, and there are few of those. So that's really where statistics is needed, because otherwise you don't get very, very far. So as I was saying, we work with this consortium, and we're particularly interested in this bleeding and platelet. Platelet is coagulation disorder. So this is how these patients are characterized. The phenotype, the complex phenotype, it's a series. First is, say, is there an abnormality, not very interesting top note, then what organ system does it concern? So is it blood or is it something else? So for example, so it's got here, it's blood and blood tissue. Then which type of cell? Is it the thrombocyte, the platelets, or is it other type of cell? What are the symptoms? Bleeding. And how is the bleeding? Is the bleeding, do you hemorrhage when you go to the dentist? Do you hemorrhage naturally? You do, and so on. So there's this kind of detailed, agreed characterization, finer and finer. And this is how you characterize a patient, that little tree, which has a structure because the terms are, if you have a term, then it is, you characterize successively downward, more refined. And this is, for example, and it's this ontology is called the human phenotype ontology, and it is like a graph. It's a directed acyclic graph, which relate this phenotype abnormality through this relationship going down. Now you don't need the whole of the tree, basically, to keep. You just need the bottom, the minimal information is just this bottom level, these bottom nodes. This minimal set of terms which are highlighted here in red. So for example, prolonged bleeding after dental extraction, after surgery, and so on. You don't need to say that this is an anomaly of bleeding if you know these bottom nodes. Now, to show you that for people who have this platelet disorders, there's many other terms which come in. Typically, there's some, they can also be coded for anomaly of their eyes, anomaly of their respiratory system. So there's many, it's very varied and very heterogeneous. But these other terms are also important because, there's some weird things going on. People don't understand why it can affect both the blood and the eyes, but it can. And this is important to find patients which are similar to each other. And so we came from the idea that we tried clustering we tried first clustering these trees, and direct clustering and then linking to association. Unsupervised somehow didn't work. There was just too much heterogeneity and things. So we thought, well, we need some kind of borrowing of information, but it needs to be in a way supervised by finding the variant as well. And so we came to define basically what we've called this phenotype similarity regression, where we construct a measure of similarity between these trees, and we build a flexible regression model to find combination of terms which are going to be shared by patients which have a rare anomaly in a gene. So in a way, we are trying to search for terms which are shared by which are shared by patients which all have a particular anomaly in a gene. Now, and in fact, as you could see, modeling these trees as statistically, as a process would be highly complicated. So what we've done is because we're interested in association and just finding this rare disease, we've decided that these trees will be our predictors, and we're using existence or not of a rare variant in a gene as our outcome. So we're using this inverse regression framework, which is much easier to treat these trees and their similarity as predictors than to try to model the tree as a response. And since we're only interested in association, then it doesn't, that will give us. And so basically, our model compares our method compares two models for the data. First, it's saying for a patient which has a rare variant in one gene, for example, at least one or two or some kind of, which we call a case, is the probability of having that rare variant at all linked to, is it just random, or can it be predicted by some similarity, by some terms which are similar across by some set of terms which are closely related to patient being cases. I think this diagram explains it better, what our model does. I haven't told you how we'll define the similarity, but assuming that you can have a distance between these terms, these trees of terms, then what we're trying to do is find this latent set of terms, so a few terms which are closer to the cases, which are there here in black, than the controls, i.e. all the others. Of course, you need controls. You can't just have cases like, And so we're trying to find this latent set of terms, which I've called phi here, and the length of the edge represent the similarity. So it's kind of moving, you're just moving among the set of terms and then seeing whether some latent, simplified set of terms will help you explain the case and control. So I don't have time to go into details, but basically, as you saw, terms are more informative if they're rare. So if two people have anomaly of their blood and thing, it gives you no information. So when you've got this database of term, the information content of the term is related to how rare the term is. So that's one way of measuring the distance. And you're trying to find, encourage people to share similar terms, and between the latent thing you're finding and the observed. And you're trying to want also that this latent thing is sort of sparser, because there's a lot of extra terms in the patient. So you basically don't want to penalize a patient for having this extra term in terms of similarity, but you want this latent set to be sparser, basically, than the other. So basically, it's based on the information content and some measure of overlap of terms. But whatever measure of similarity you come from, there's absolutely no reason that it should be linearly influence the probability of having, yes or no, a rare variant in a gene. So you also need a flexible, so it's a kind of non-parametric, so you also need a flexible transformation, so that whatever distance you've defined, you've got a kind of, so if we've used a beta cumulative function with different parameters or something like that, to actually link basically this similarity measure to the probability of having or not the rare genotype. And so, we're going gene by gene to actually do that. So we're defining for each gene whether there is or not, for example, at least one rare phenotype or not, one rare variant or not in the patient. For this patient, and this will be a case y = 1, and otherwise no. So this is the idea of trying to first, the main idea is use this inverse regression, define a measure of similarity which is meaningful, but model its effect in a flexible way, sort of non-parametric way, because you just don't know. So you don't know how it's going to influence. And we've used basically a model choice. So we're running a Bayesian algorithm which just selects either it's the model with where there's no link at all, or it's the model where there's a link. And we're interpreting the posterior the posterior mean of this model choice indicator there, gamma. No time to detail the price we've used. What's important is that you've got a prior on the set of terms, and if you have some information on, from, for example, a similar mouse model or something, which is very important because, you have very few cases. You want to actually get as much additional external information as you can, then you could actually use it in your prior on the model space of these terms. And we find that absolutely very important. We've applied that to data on 2,000 individuals. There was only rare variants, And so basically, as I said, we're going through all the genes, one at a time, and a different model of saying what's the case could be at least one rare allele in the gene for a dominant model, at least two rare alleles in the gene if you're looking at rarer recessive model, or if you have other classification of your rare variants, you could use it to define different definition of cases. What was very useful to us was to have a kind of a few training example of where the variance was already known, and if we weren't able to recover that, then our method was worthless. And that really helped us to actually put enough flexibility in this non-parametric function and so on. So we did use a training set where we knew we should find something. And in fact, for these gene, actin N1, which is a gene of the disorder of the platelet, where the platelets are big, then we had quite a reasonable sample size with basically 43 individuals carrying that, of which 22 were coded, and we found that very the probability of choosing the model was 1, that there was influence of that gene, and that here it's only displaying the typical tree, but the kind of the marginal probability of the terms. And in fact, it was a combination of two terms, thrombocytopenia, so not enough platelets, and having these platelets with a bigger volume, which was the most characteristic two terms. And when you run that on the whole of the database and all the gene, we came up, so here on this plot, you've got basically just the probability of having an association. And you could see most of the time it was 0, some of the time it was 1. And here in red are those for which there's been confirmation, validation that these findings, these variants, have been validated as being functional. Some of the, for some of them, this black bar is like new discoveries which need to be validated. And as you could see, it's quite a lot of these gamma equal 1 have been validated, and so it just shows that the method is capable of recovering what is known, and it's capable of discovering new things which can be exploited. Angela, how long have I got? 5 minutes? Okay, so very briefly, I will just talk about, without giving any detail, but the third area where we're trying to look at basically when you have a large number of molecular features and you want to see whether they're conditionally independent or not. And here is Gwena Elodi, who has developed this work, and he works in the BSU. So sometimes when you're given a large number of features, the first pass is to see what's linked to what, but because there's a lot of indirect link, it's very useful if you could actually test the conditional independence. Are these things, when you condition on the whole of the feature, are these things linked or not? And Gaussian graphical models obviously are, Gaussian is always an assumption, but they provide a very key tool in that area. Now, there's been a lot of work on trying to search for graphs, and these graph of conditional independence, and this is not scalable to very large number of features. It's notoriously really difficult to explore this graph. So basically, when we talk about the graph we recover, it's in fact, we're testing each link, so we're testing through an explicit computation of a base factor, which got an explicit expression. We could test for the existence of this link or not. And once you've done that, you actually can reconstruct a summary graph, basically, of conditional embeddings by putting all the links after having applied some multiplicity correction. So it's a very simple idea. It's first, we're not exploring fully the joint distribution, but we're testing each of these edges, and then we've derived also an explicit distribution under the null, which means that you could actually compute the p-value, and you could use whatever multiplicity correction that you want to actually show the graph of all these links corrected for multiplicity. So maybe, so the first thing to do is you need to estimate the precision matrix. And to do that, because you've got n, which is much smaller than your number of nodes in your graph, you need some shrinkage estimator, and we've gone for the simple linear shrinkage, which is basically implemented through using the conjugate distribution. So you've got basically your y's matrix, a normal, and the sigma, which links basically the nodes, is given an inverse Wishart with degrees of freedom delta and prior scale matrix D. And then you've got the whole framework, the posterior distribution, because it's conjugate. They've got this ninth form, either inverse Wishart for sigma or for the precision matrix gamma of interest Wishart, which involve, of course, the data S, the sum of square, and it involves the prior, so that you have actually got basically positive definite expressions for the posterior summaries, either for sigma or for the estimation of sigma, so shrunk estimate, which is positive definite, or for the precision matrix gamma. So this is simple, this is fast to do, and if you had some information on D, then you could take the identity typically, but if you have some information on D, you could include it. Now, in terms of the testing, what you want to do is test, as I was saying, the existence of a link. Is it zero or it's non-zero? And we inspired ourselves from previous work from Paolo Giudici, which had proposed closed form Bayes factors for this problem. However, the expression that he had derived involved the inverse of the precision matrices and was not computable. So in our setting, so we derived another expression basically for the Bayes factor, which involves very simple, which has a very simple intuitive explanation. It involves the prior partial correlation and the posterior prior correlation, and this is now which can be computed with our shrinkage estimate. And the Bayes factor is consistent for selection and so on. And not only that, but you could actually derive basically under the null, the tail probability follows, is linked to a beta distribution. So you've got a computable expression, and you don't need to use permutation to actually derive the tail probabilities. You could get them through the beta distribution. So you could see that it's a very fast way of performing that and recovering the graph. And when compared with other methods, we basically, in terms of operational characteristics, it's good. It's as good as the others, but it is much faster. So I will stop now. I won't have time to show you how we use that to look at the structure of this hotspot I had covered. I just want to go on just my final slides with concluding remarks. So basically, enriched response model, what everybody is now working on. It can involve multiple quantitative traits. It can involve finding meaningful distance between responses. As more and more layers of information, including image data and so on, will be derived on this for the same patients, there is a lot of work for developing integrative models which could make use of these data. And maybe having a first pass at this as a network, finding features which are associated gives you a group of features to model, and then going in more depth there is one strategy. There's substantial computational challenges, so it's actually worth thinking of approximation algorithm exploiting conjugacy and deriving closed form expression for some part of your calculation if you want. And again, here I acknowledge all my collaborators, which I've enjoyed immensely working with, and a few references. Thank you very much.
Well, thank you, Silvia, for your very interesting and stimulating talk. Unfortunately, we are a little bit late, so we haven't time for many questions, but for just one, if you have. Okay, two, and then we must stop. Okay, please.
The point of this, I guess, is to look for targets for interventions to try and stop the diseases or decrease their severity. But for that, you need to have some causal relationships. Can we assume causal relationships here, or is that an additional piece of complexity that you have to introduce into this modeling?
Well, so, I mean, one of the, between the DNA, the DNA, you know, for some of the things, you know how the flow of information goes. You can't remodify your DNA. So for some of the models, somehow you could assume some part of directions, but I agree with you, there is, as you go further down the chain, there could be definitely inverse, some modification and so on. So I think once you want to actually target treatment, and you need to have already found what could be the likely feature that you want to model, and then you need to model more precisely. So people use you use also the genes as instrumental variables. You could try to get experimental work where you interfere, where you actually intervene so this is still hypothesis generation, quite refined, but then you need to confront that by experimental work. And this is why I said we're not focusing so much on prediction, but really on finding the few key features which can be modified, animal model and so on. But also trying to try to find the direction of the arrows. It's sometimes possible that, because you've at least got a route. If you have the DNA, you've actually got a route, and some arrows have to go that way. But as you go further down, no. But it can help you to decipher at least a few of the plausible, directed things.
Mauricio?
Just a question to understand the similarity in the second problem.
Yes.
Is the similarity connected with the correlation between responses and, why similarity?
The similarity is, because these are categorical terms, it's connected to the sharing, basically, of terms. So in a way, it's a kind of correlation. But, and the content of the terms, i.e. how frequent or how infrequent they are. So basically, it's a kind of intersection measure. But it becomes quantitative because then you look at the intersection, and then you look how rare these terms are, and it's like inversely proportional to how they're rare. So but the way we're using it, a regression, you could have done like a Boolean type regression, but it's this hard things doesn't work very well. There's too much heterogeneity. So we try to make a continuous, transform that as this continuous measure of similarity. We started with a Boolean type of logic regression and tried, but this didn't. There's too much heterogeneity, and sometimes there's too few. It's not clear-cut enough for this to work well. It was difficult to interpret, but it's this idea, but transform more continuously. But to define that similarity, you really need to understand your problem. And I think it's got wide applicability in other areas if you understand really the structure of your problem, and you could define meaningful measure of similarity, you could discriminate, then for very different problem, I think this is could be a useful tool, sort of non-parametric type.
Well, I'm sorry, we must stop the discussion here because we have other sessions. I'm sure Silvia will be happy to answer all the questions during the breaks today. So I thank her again for this very nice talk and all of you for participating.